The next speaker is Dr. Sihem Boudina, who is a professor in the Nutrition and Integrative Health Department, Integrative Physiology, I'm sorry, department in the College of Health.
College of Health, yes. Okay, well, I will just start by thanking Alfred and team for this invitation. I didn't know that this existed for 10 years, so this is new for me, and I love it. All right, so this is a good layout from Colleen to me, because I'm going to be talking to you about a transcription factor called PRDM16 that has been involved in thermogenesis. But in the context of today, we're going to really talk about sex differences in a cardiomyopathy that we basically discovered. So I'm going to talk about a genetic disorder called 1p36 deletion syndrome. So it's a chromosomal deletion. It's actually the most common chromosomal deletion in humans. It affects 1 in 500 individuals, and mostly kids show up with a lot of phenotypic manifestations, including some of the ones that are highlighted here. But for us, I'm a cardiac physiologist, so I'm interested in cardiomyopathy and how cardiomyopathy develops. And we stumbled on this project, I would say by accident, because my postdoctoral fellow, Carla Maria Perez, was writing a review on PRDM16 and thermogenesis. And she came back to me and she said, all what I can find in human, I said, is there any mutation in human that you really show a body weight phenotype? And she comes back and say, no. All what I can find is that human who carry PRDM16 mutation, 25 to 35% of them actually develop cardiomyopathy. And I said, huh. So that's really when we started really embarking in this project. And we do a lot of mouse genetics. So for us, it was really easy being in the same building as Mario Capecchi to use the LOXP system and create recombinase to generate a PRDM16 knockout in the heart. And that's how everything started. So PRDM16 is, again, a transcription factor. Its structure is pretty much, I would say, conserved between mouse and humans. In humans, they have 2 isoforms of PRDM16. In mouse, they have only 1. And the gene has been really related to leukemia and has been really studied in the context of leukemia for a while. But we didn't know what this gene really, does. And I think in the last 10 years, we really came up to really understand that it plays a big role in differentiation and maturations of cells, variety of cells, and brain cells, but other cells as well. There was a study from a Dutch group a long time ago that showed that if they take patients with 1p36 deletion syndrome, and if they stratify them and look at the genes that they lost, because remember, 1p36 deletion syndrome is mostly affect the short arm of chromosome 1 that is lost, and within that deletion, you can lose many genes. Okay. And what they found is that if you happen to lose the PRDM16 locus in those patients, you're most likely going to have cardiac manifestations. And what I said by cardiac manifestations, it could be like a congenital heart problem or a cardiomyopathy or a non-compaction cardiomyopathy. So based on this study, we started a collaboration with a physician scientist at Duke University. He's a pediatric cardiologist, and 2 individuals, so one from his lab, Ryan Kramer, and one from my lab, who's now an MD PhD here at the University of Utah, Nima Fatahian. We started this wonderful collaboration where they collected a cohort of 1p36 deletion syndrome over 5 institutions, including the primary children here. And they built that cohort to look at specifically whether the deletion of PRDM16 can cause cardiomyopathy. And what they found is, oh, too close, okay. So what they found is that if you lose PRDM16 here, you can actually develop 3 times more cardiomyopathies, okay? And if we divide those cardiomyopathies in subtypes, we found that it's mainly non-compaction cardiomyopathy, but also dilated cardiomyopathy, but not hypertrophic cardiomyopathy. So it's very specific to these 2 cardiomyopathies. And then if you happen to lose PRDM16 with this deletion, you're more likely to die, and your freedom of death is actually really reduced when you lose PRDM16 within that deletion. But what was astonishing for us is when we separated the sexes, we found that first of all, females in general are more affected by 1p36 deletion syndrome, and we have no idea why, okay? So they are more represented. But most importantly, even if we take males and females who have lost the PRDM16, right, locus, females do worse, okay? With the same deletion of PRDM16, they do actually worse, okay? And they develop twice as much, or they have twice as much incidence of cardiomyopathy than male who lost PRDM16, okay? And of course, I'm not a physician, so I went to genetic in mice. And for me, it was really gratifying to find a mouse model that can actually reproduce the human data, because it's very rare in science when mice are different than humans. So in this model, what we did is we targeted the deletion of PRDM16 in cardiac cells. And what we found that if you do that, mice die with dilated cardiomyopathy within 5 months, okay, of age. And when you look at their heart, it's full of fibrosis, and they have an increase in all of the pathogenic genes that are normally associated with cardiomyopathy. But then if you look at their survival, and we separated males and females, so this is the global survival, so they die sooner. But if you look at who is responsible for this death, is mainly the females. So the females collapse much sooner than the males in this knockout. Of course, that intrigued us to look at specific pathways. So what we did is we actually took these mice even before they developed cardiomyopathy, and we screened for their genes to look whether we can come up with some pathways that are differently regulated in males and females. And sure enough, you can find that some common pathways are represented in both males and females, and those are usually the pathways that are known to be regulated by PRDM16, mainly metabolism, I would say, and oxide phosphorylation. But then there were some pathways that were unique to females that were really dysregulated when PRDM16 is lost. And those in particular are mainly, I would say, autoimmune responses, DNA damage responses. Those were very unique to females' heart compared to male hearts. And first of all, we went with the easy path. We said, okay, metabolism is very important in the heart. Can we target metabolism to reverse this cardiomyopathy, since we find it both in males and females? Okay, so we focused on a particular gene that was actually downregulated all the time when PRDM16 is lost, and this gene is called PERM1. So PERM1 has been really involved in regulating metabolism in skeletal muscle, but also in the heart. And we said that, it's not very easy to target PRDM16 because PRDM16, both in humans and mice, is expressed in a very short period during development, and then it goes away. So if you target this gene by gene therapy, it's impossible because you don't know exactly when you're going to give it. Whereas PERM1 was a perfect candidate because PERM1 is only expressed in muscle, including heart and skeletal muscle, so it's very specific, but it's also involved in metabolism only. So we said, well, maybe this will be a much better target for us if we want to do that. So we use gene therapy, we use AAVs to deliver one injection of this PERM1 plasmid. And again, we confirmed that we can actually increase its expression. We used the GFP AAV to look at that. We infected pretty much all the cardiac cells, and we increased both the protein and the message of PERM1. And surprisingly, when we did that, we found that we can actually improve. Look at the females first. Okay, so when you look at the females, we know that these females develop, again, they have reduced ejection fraction at about 20 weeks of age, and this increases with PERM1 delivery. Same with fractional shortening, and most importantly, the left ventricular internal diameter, which suggests that, of course, in the knockout without PERM1, they develop dilated cardiomyopathy, so they have a dilated left ventricle. And this dilation is actually prevented when you give PERM1. We think that males are also protected. However, we think that by giving a PERM1 in a wild-type male, that's not very well tolerated. So a wild-type male should not actually receive PERM1, and when they do, they actually behave in a different way. It seems like it doesn't protect them, it makes them worse. But if you look at just the knockouts, again, the knockouts that received PERM1 seems to do a little bit better in terms of dilation as well. So, and that's, of course, when you see this data, you always go to your grad student and you say, show me the raw data, because I don't believe it. However, when I saw this data, I start really believing it, because you can probably misinterpret echocardiography images, but you cannot really make the mouse live longer. So this is just showing the data for the survival of the females, and the ones that received the PERM1 AAV actually survived. Of course, this is up to 20 weeks. Now we are doing it for longer to see how long we can actually extend their survival. So why is this important? These 1p36 deletion syndrome patients, right now they have three options. If they progress into dilated cardiomyopathy, they are given heart failure medication. And both males and females, they are just giving it blanket heart failure medication. We don't have any data to suggest that whether you would prevent their dilated cardiomyopathy, and whether females actually have a much better diagnosis. But most of the time, because they have this dilated cardiomyopathy when they are children, they have to wait for a transplant, because you cannot keep them for a long time in this medication. So there is a need to develop a new therapy for this population. And we hope, I just actually filed a provision patent for this PERM1, and hopefully maybe we can do more human data to show that this is going to be helpful. And with that, I will just conclude, and thanks the people who did the work, most of the work. And I'll take any questions.
Is Dr. Yamaguchi here? Oh, in that case, take your time. Please go ahead.
Okay. Yeah, so I didn't show that data here. There's another side of this, which is really just specific to females, that we are now digging. So what we think, and again, we have no idea how this happened, but what we think that there is maybe some epigenetic modulation, because PRDM16 has also a DNA methyltransferase in its structure, so it changes the chromatin. So we think that there are some epigenetic differences between males and females that leads to more DNA damage and less DNA repair early on in the females when they lose PRDM16. And that actually triggers the STING pathway, and that triggers the immune response that we see. This is a sterile inflammation, so this is more of an interferon gamma response. Yeah. Yes. Yeah. Yeah. Yeah, we haven't done it because, of course, this is a cardiomyocyte-specific promoter. But now there was, I think, a new study that just came up that showed that um, PRDM16 is actually very highly expressed in smooth muscle. So it is possible that, it's probably doing something to smooth muscle cells and, changing the vasculature as well. Yes. Very good question. So we did overectomies. We didn't do VCD, but we did overectomies in these females, thinking that maybe, like we can make them worse because they lose, It actually, we think that this is independent of estrogen. So when we did overectomies, we didn't, they still developed this dilated cardiomyopathy the same way. So again, this is us thinking that maybe this is not really related to hormones. No problem.
Okay, if
All
not,
right.
thank you very
Thank
much,
you.
Sihem. Thank you. Dr. Yamaguchi.